Principles of enzyme catalysis. Catalysis is the process through which the rate of a reaction is altered while the quantity and chemical properties of the catalyst remain the same. These reactions usually show an increase in the rate of the reaction with the help of a substance called catalyst. The catalyst is not used up in the reaction and can be used repeatedly. Usually, a small quantity of catalyst is enough to speed up the chemical reaction. In most of the reactions, the catalyst reacts to form a temporary intermediate which further regenerates the original catalyst. Now, what is enzyme catalysis? Enzyme catalysis is the process by which there is an increase in the rate of the reaction through a biological molecule called enzyme. Catalysis by enzymes in a cell is vital because most biochemical reactions have meager rates of reactions when uncatalyzed. Therefore, for a reaction to be successful, the molecules of the reactants should contain sufficient energy to cross the energy barrier, that is, the activation energy. Enzymes function by decreasing the activation energy by forming an intermediate enzyme substrate complex before the release of the product. Principles of enzyme catalysis. Enzymes are a type of catalyst used to modify the rate of reactions. These are also called as biochemical catalysts. The principle of enzyme catalysis is that the vital factor is the reduction of energy, which acts as a barrier between the reactants and the products. One of the most important principles is that enzymes always catalyze reactions in both directions. Reducing the activation energy increases the number of reactant molecules to overcome the barrier for product formation. Hence, enzymes cannot drive a reaction forward or affect the equilibrium position. They can only increase the speed of the reaction. Looking at the characteristics of enzyme catalysis. A single molecule of the enzyme catalyst can transform up to 1,000,000 molecules of the reactant per second. Hence, enzyme catalysts are said to be highly efficient. Secondly, they are unique to certain types of reactions. That is, the same catalyst cannot be used in more than one reaction. Thirdly, the effectiveness of a catalyst is maximum at its optimum temperature. Next, the biochemical catalysis works best at an optimum pH between 5 to 7. The activity of the enzymes usually increases in the presence of a coenzyme or an activator. The activity of the biochemical catalyst declines at either side of the optimum temperature. Let's have a look at the molecular basis of enzyme catalysis. The entire process begins with the presence of a free enzyme which undergoes the process of substrate binding. This means that the enzyme binds to a substrate at the active site, forming an enzyme substrate complex. Further, during the catalysis reaction, there is a formation of an enzyme product complex, which in turn undergoes few changes, conformational changes, to bring about the release of the product. Along with this, the free enzyme is also released so that it can once again get attached to another substrate molecule and the whole process continues. Let me explain more clearly. Firstly, the enzyme randomly binds the substrate, allowing to form an enzyme substrate complex. The enzyme bound substrate now transforms into a transition state of the reaction. In the next step, the enzyme also undergoes conformational changes to accommodate the substrate, creating a more favorable connect of the substrate and the active site. This is also referred to as the induced fit model of enzyme catalysis. The enzyme stabilized transition state undergoes further changes to become enzyme-bound product. This complex then dissociates to form a product and a free enzyme. In the last step, the released enzyme is now ready to catalyze the conversion of another molecule of substrate to the product. Hence, it is to be noted that the enzyme does not change during the reaction. It remains in the same state as before participating in the reaction.